Okay, guys. Sorry, the roller coaster ride is over. It was nauseating, wasn't it? Yeah, it's funny. I don't know. This is just going to be. This is actually my first time ever lecturing this many technical difficulties as I've had. It's like, what is it? It's my third semester here. No, fourth semester here. And maybe like fifth or sixth time teaching. And I've never had this many technical difficulties. So it'll be fun. Okay. What are we talking about? Chromatin structure and function. There we go. Okay. Is that what I put first? Huh. Okay. So what is chromatin structure and function? Because sometimes these biology teachers say things and it's just these words that have no meaning, and you're like, I don't know what that is. Hopefully I'll figure it out by the end of lecture. So let's start out with understanding this. What is the meaning of chromatin structure and function? Someone just take a guess. You don't need to have the right answer. You can say the completely wrong thing and I'll just say no and we'll move on. It's important to get used to people telling you wrong. Yes? The shape of the chromatin and why it's in that shape. Okay, let's add to that. Yes?
How the shape relates to the function.
How the shape of a chromatin relates to the function. Okay?
What makes up all that and how it works.
What makes up all that?
Yeah.
Meaning the shape
Yeah, sure.
and structure?
Sure,
And how it works, the function. Okay, we're giving a lot of great definitions. What do we mean by structure, though? Do you mean the double helix? Yes.
The way the DNA is arranged around the histones.
The way the
relate to each other.
right. So the way that the DNA is arranged around the histones, okay, and how they interact, how the histones interact, also, right? What else?
How they lay.
Okay, so how the histones fold onto each other to compact. Okay, so we're getting better with, we're getting more and more specific with structure, right? So whether, you could say whether your DNA is open or closed, right? And how does that relate to function? What do we mean by function? Yeah.
Whether transcription can occur or not.
Right, so that's one function, whether transcription can occur or not, right? Because if your promoter is wrapped tightly around a histone, which is then folded upon another histone and all compacted, you'll never have transcription of that gene, right? What else? Yes? Whether replication or repair can happen, right? So you're starting to see it has to do with accessibility to the DNA, right? So whether or not different processes in the cell that require accessibility can happen. So yeah, to repair DNA, the repair enzymes need to access the DNA, right? It's not like a magical blessing that's sent from afar. It actually, these enzymes and proteins need to bind to the DNA and do what they need to do to fix it, right? Anything else? What about, how does chromatin structure affect the function of mitosis? Is there anything that change? Is there some kind of chromatin structure associated with mitosis? I'm hearing. I want. Huh?
DNA is packed into chromosomes.
DNA is packed into chromosomes. Yep. Sister chromatids are attached. So sister chromatids that have to do with you've gone through replication and you're attached, right? And then you do have, so I saw another hand. Yep.
It has to be condensed.
Right, you need to have condensed DNA. You don't want your DNA all unraveled like a mess of spaghetti, right? Because what happens if you segregate your chromosomes and it's not condensed? Just imagine pulling apart a big mess of spaghetti. What usually happens to that spaghetti? It falls apart. What else happens? It breaks, right? So do you want to break your DNA? No. Broken DNA is a bad thing. We could probably all agree. So that's why you need to condense your DNA into chromosomes before. So that actually has something to do with the structure and the function of chromatin, right? Okay, so you guys all had really great examples. So I put this picture up. And so for those who are still kind of getting into the swing of things with this class, chromatin means DNA and the proteins, which means histone and non-histone proteins. What are non-histone proteins? That might be in chromatin. Yes.
Histone acetyltransferases.
Okay, so histone acetyltransferases, so proteins that maybe function in doing something with DNA. So one would be, or histones. So histone acetyltransferases, which we'll learn about. What else? HDACs. Yep. So that they deacetylate. What? Do you have another one?
I was thinking just about like polymerases, so that we can, when we start transcription.
Polymerases. Well, would they normally be associated with chromatin, or maybe only during certain times of the cell cycle? Yeah. Yeah. So I guess they could count, but I wouldn't call them as part of chromatin because it's very limited to S phase. You're not completely wrong. Yeah. Nucleases. Oh, do you want nucleases associated with your chromatin? I guess if you're repairing DNA, some proteins with some nuclease activity might be found. But normally you really want to not degrade your DNA. Yeah. What about transcription factors? What about nucleosome remodeling complexes? What about all these proteins that are going to post-translationally modify histone? So we heard histone acetyltransferases, methyltransferases. So just starting to think about what might be there. Yes?
Are these
Transcription
transcription factors present?
factors will be present bound to the DNA when they're needed. Not all. Well, we'll have to think about it because certain repressors might be bound quite frequently, right? So, and that would be that might not be necessarily transcription factor, but really more repressor. I wouldn't say anything's going to be bound all the time. You have to think of your chromatin as something that is dynamic and constantly changing. Constantly changing because you've constantly got different genes being expressed. Yes?
So even while the DNA is wrapped around, you can still have a bind to the DNA as a
No. So the question was, while the DNA is wrapped around histone, will you have a transcription factor bound to the DNA while it's wrapped around? And that's a great segue into what this picture is showing here. In order to get transcription, you need to open up that region of DNA to expose your promoter or any enhancer regions so that transcriptors can come and bind. And you'll have nucleosome remodeling complexes do that. And we'll learn more about that. But so here, this is just showing the different ways that chromatin structure can change, right? You can have an inactive conformation. So here you've got your DNA wrapped around your nucleosomes. And notice you've got your promoter, that's TATA binding protein, that's where your TATA binding protein is going to bind. That's wrapped around your histone, so your TATA binding protein is not going to be able to bind. And so a nucleosome remodeling complex would have to come in and slide the histone away to expose that site. And this is just showing how you'll get recruitment of chromatin remodeling complexes, and they'll slide around the histones to expose the binding site, which allows this whole transcriptional activation complex to start to assemble. And that's how you'll get gene expression. So big picture, we're starting with the big picture. Don't get bogged down with all these little details. I just want you to think very, very large here, right? You've got this chromatin, and it's dynamic, it's changing its structure, meaning how densely packed or how much it's open, based on whether or not transcription needs to happen, what part of the cell cycle you're in, whether you need DNA repair to happen because there's damage. There's all these things that are affecting the structure and the function of chromatin. Any questions before I move on? Yes.
So I was wondering, what's the purpose of the transcription factors being included in the chromatin?
What is the purpose? Thank you. What is the purpose of transcription factors to be included in chromatin? Who wants to try to answer that one? I know it's my job to answer, but I bet you someone in this room has an idea why transcription factors might be bound to chromatin. Okay, well, how does gene expression happen? You all took 309. Who did not take 309? Okay, because I'd have to. You didn't take 309? Okay, that's going to be bad. That's really bad. You should be in this class if you haven't taken 309 because you're going to really struggle. Okay, so or 309 equivalent maybe elsewhere. So you've all learned about transcription and translation. And for transcription to happen, saw a hand up over here somewhere. Yeah, you were going to add something, right?
Yeah, I was just going to say that if the transcription factors are closer to the DNA, when a signal is received, it'll be like more efficient if it's closer than if it were far away from the DNA to transcribe the region.
Okay, so what I'm hearing is you want your transcription factors to be close. Well, as you remember, what we learned in 408 is transcription factors are usually kept out in the cytoplasm until they become activated by signaling pathway, which exposes a nuclear localization signal so they can go in to the nucleus. So you're thinking about them in terms of maybe they're bound to a histone or something like that. No, they actually don't come in until they're needed. But that's why they would be bound to chromatin. They have to come in and bind to the DNA at the promoter region so that transcription can happen, because that's how they recruit RNA polymerase and all the transcription factors. Well, basically with the goal of recruiting RNA polymerase so transcription can happen.
Okay, so that means that transcription factors is not part of chromatin.
They are recruited to chromatin. So you have to think of chromatin as something that's very dynamic and constantly changing. The only thing you could say that's always bound are histones, right? And they're even moving around. Yes?
So chromatin is the DNA in any proteins that are currently associated.
Yes, you can.
So if it is S-base and DNA polymerase is bound to it, then that would count as part of the chromatin in that instance.
Yeah. So like when you do a chromatin IP, what you're doing is you're immunoprecipitating a protein and any DNA and protein that's bound to it. So that's the way you should think of chromatin. And frequently, if you want to prove that a transcription factor or something is bound to a protein, you'll do a chromatin IP with an antibody against that specific transcription factor. Yes?
The transcription factor only binds after the DNA is unwrapped from the histone.
Right. The transcription factor is only going to bind after the DNA is unwrapped from the histone because it needs to access the actual DNA, that sequence of the DNA. And that's where regulation comes in, right? And so what we're going to be learning about is how you can move these histones around and control the structure of your chromatin to regulate what genes are being expressed. So you're going to expect that during certain phases of the cell cycle, certain parts of the chromatin are going to be open. You're going to have lots of transcription. During other phases of the cell cycle, things are going to be really condensed, so you won't have any gene expression or limited gene expression. Different cell types are going to, their chromatin is going to look different because different cells, like an egg versus a neuron, are going to express a very different set of genes at any given time because they have different jobs. So, different structure of chromatin. Any other questions? Are we understanding the big picture? Should I move on and get into details? Or should we just stay with the big picture? Skip the rest of the lecture. Okay. Okay, so is the chromatin structure the same or different in these two cells? Different, right? You say different. Here, would you say it's tightly packed or loosely packed? Tightly packed. Because what is this? What are we seeing here? So you've got chromosomes being pulled apart. What phase of the cell cycle is this cell in?
Interface.
Good. What phase of that cell? What phase of the cell cycle is anaphase in? Mitosis. So this is a mitotic cell. Highly condensed chromosomes, DNA. The chromatin looks very different than in this non-dividing cell, right? So would you say, where do you think transcription is happening at a higher rate? And which one?
Actually, no, the non-dividing.
The non-dividing cell, because everything we talked about is how the tightly packed DNA, you can't get transcription factors in there, you can't get RNA polymerase in there, you can't get any significant gene expression when you've got your DNA very condensed. So you're going to have much less gene expression during cell division. I'm not going to say none because I don't like to be definitive about anything. Okay. Yes.
At that point, when the cell is dividing, don't you want there to be less transcription happening anyway? Because at that point, this is supposed to be the same when they divide. So wouldn't that cause problems if it did continue to?
So wouldn't you want less transcription during cell division? Because you want your daughter cells to be the same. I guess so. Also, it just doesn't seem to make any sense to be doing lots of gene expression while you're dividing, does it? Just doesn't. It seems like your resources should be towards separating those chromosomes effectively, right? Because if you don't do that well, then it's not good. Okay, so why did I write this? How? How is the structure different? We talked about that. How does it relate to the function? We talked about that. Okay. Any questions about this concept? We all understand this concept, right? Everyone feels good. Are you ready for a test? Why not, right? We haven't learned anything yet. Okay. Okay, so I'm going to just harp on this for a little bit. Chromatin structure changes throughout the cell cycle based on what is happening at each point of the cell cycle. I want you to really understand this is dynamic. It's in response to what the cell is doing at the time, right? So you'll have, during interphase, you have this open chromatin, more open chromatin, you'll have gene expression and you're having chromosome replication. And then as you go into M phase, you're going to have a condensed chromatin, so not really any significant gene expression. You'll have cell division, and then you'll have this more open state of chromatin. One day I'll figure this thing out. Again, here's a close-up to what it looks like. Mitosis, that's really, really condensed. And this is an interphase, right? Really open. You can see actually individual beads on a string, kind of, if you look real close. Yes?
So if a pathogen were to attack a cell as it's entering mitosis, it wouldn't really have a chance to defend itself.
If a pathogen were to attack a cell, yeah, a virus of something were to infect a cell during mitosis, would the cell have a chance to defend itself? Who wants to take a... Now that's a really nice question, because you have to think about a lot of things, right? You have to think about a lot. Well, let's see what the initial answer is.
Isn't that when we use vectors to get into cells when they're divided?
In terms of transfection?
Yes.
Do we transfect at cell division? No, you can transfect cells at any time. It all depends on your method of transfection. There might be higher efficiency during cell division due to the lack of a nuclear envelope. I know some viruses do prefer during cell division and actually will arrest the cell cycle to help infection because they can't get into the nucleus, like HIV does that. Yes?
Are there any instinctual defenses that a cell has to kind of like keep its objectivity during cell
Okay, are there any defenses the cell has to keep its guard up? So I guess the thing we need to think about is how long is mitosis? Really short, right? It's fairly fast. So I would say, so to go back to this, first of all, is I would say probably yes, maybe a cell is more susceptible to not being able to do transcription and translation in terms of mounting an immune response during mitosis, but it's a really short period of time and you need to think holistically because don't forget, before the virus gets into the cell, probably the virus has gotten into other cells, and those cells are now secreting cytokines so that they're already, like, the whole surrounding area is flooded with cytokines and these signaling molecules that have alerted the cells to the fact that there's a virus around or a virally infected cell or some kind of pathogen. So they've had a chance to start to ramp up an immune response anyways. That's one thing. The other thing is, let's say you're the first cell that's being infected. There is the innate immune response. So there are ways that don't require lots of gene expression. And then there are also these other mechanisms for protecting the cell, like the cell does not like to see double-stranded RNA in the cytoplasm, and that often comes from viruses. And so there are mechanisms in the cell to degrade that. So if a virus is uncoding in the cytoplasm, the siRNA system will come and attack that. And then there's a whole bunch of different mechanisms. So it's not just one, it's not just dependent on gene expression, but the gene expression will increase over time. So you always have to think of a cell as part of an organism and there's this communication happening. You had a question?
Yeah, also I was thinking, so even if the virus were to get in, there's no nucleus and no euchromatin for it to do anything there.
So if the virus were to get in, there's no nucleus and no euchromatin. Well, the thing is that all viruses are different, and sometimes you don't need much of anything. Some huge, like herpes viruses, tend to bring in a lot of what they need. The other thing to think about is there DNA polymerase, RNA polymerase, all of that available at the time for the virus to replicate? There's so many pieces to that question. It's a really great question, though. We could probably talk about it for the rest of the class. It's a good question. Anyone want to add to this discussion? Okay. Good. It's good to think about things. Thinking is important. Okay. So here's your condensed versus decondensed chromatin. Here it's condensed, so it's kind of, this is where the nucleosomes kind of fold up onto each other. And then here it's been unpacked. Here, so nucleosomes are the histones with the DNA wrapped around, and that's what you're seeing, these beads on a string. And so this takes us into the paper that we're doing in recitation this week. So the nucleosome was discovered in 1974, and the paper that we're reading is one of those papers that came out where they were talking about how much DNA is wrapped around the nucleosome, or a histone. And this is the experiment that some of you guys, well, everyone who did it on Monday already has, and everyone who's doing it today already read the paper. Sorry, Friday people, you're getting, well, Friday people are lucky because they're getting a little tutorial before you've probably read the paper. Right, so what they did when they were studying nucleosomes is they treated the unfolded chromatin, so that's that chromatin, the beads on a string chromatin, with nuclease. So what does the nuclease degrade? What's it degrading? DNA. Which DNA? The naked DNA. The naked DNA is the DNA that, the linker DNA. So this is the DNA here, right? Why can't it degrade the DNA wrapped around the histone? Who wants to raise their hand? Yes? Cross-linking. We didn't cross-link anything yet. Why can't the nuclease degrade? Yes?
You're just not able to get in because it's so tightly bound.
Right, it can't get in. It's tightly bound. When you've got DNA wrapped around a protein, how can another protein get in there and bind to the DNA in an efficient way so it can degrade it? So that's how that works. So it degrades all this linker DNA, and what are you left with? Yes. Your nucleosome, right? You're left with your DNA wrapped around your histone. Okay, so what you do is you've treated your chromatin, your unpacked chromatin, with your nuclease. You've digested your linker DNA. You're left with this nucleosome core particle, which is your histone, with the DNA wrapped around it. And then you treat it with high salt. Why high salt? Now this is the kind of stuff that I love to teach you guys, because if you know chemistry, it's very apparent. If you don't, no one, like a lot of people don't think about chemistry in terms of biology and the how they meet up, but it's useful to understand these things. Yes?
Isn't there ionic bonds between the DNA and histone, so the salts are, they want the electrons, so it pulls it apart, like it's just
Right. So the bonds between the DNA and the histones are broken when you have a lot of salt, because the salt is pulling away the electrons. Yeah, that's perfect. And so basically what it likes to do is it likes to bind all the water. That's the way I think of it. And that prevents the interaction between the DNA and the histone. And so that this, or even DNA, histone, the histone subunits. So anytime you want to disrupt an interaction that's based on hydrogen bonding or anything really, other than covalent bonds, then high salt's a great way to do it. And that's what they did. So they treated with high salt, and they got all the subunits. So they got this DNA, because they're trying to find out how long the DNA is, how much DNA is wrapped around each histone, and they were able to get all the subunits of the histone octamer. It's an octamer, meaning there are 4 dimers that make up the histone. Yes?
I had a question. In the paper it talked about how there were 5 main types of histone, and then in the other paper said there was 2 of each kind, but there's only 8 total units.
5 main types. Well, I can tell you this for sure, is this H2A, H2B, H3, H4 are your main subunits of histone, and they exist in dimers, and we're going to go through how they assemble in a minute. There are histone variants that are frequently used, like the centromere in DNA damage, a whole bunch of different kinds of histone variants that will replace some of these, so they may have been referring to that. But they do exist as dimers that form an octamer. Yes. Yeah.
Wasn't it because, like, her question, there was one more, I think H1, it was either H1 or H5 that's
Yeah, they said that there were 5. Yeah, it says the main 4.
Oh, there's the one that's involved in binding the histones to each other, right? Yeah. Anything else? Okay. Okay, so here's the structure of the nucleosome core particle. I want everyone to memorize this and be able to draw it. I'm just kidding. Who had a moment of panic there? Okay, good. As long as you realize that was a ridiculous request. So it's showing the different, it's on the bottom here, red, orange, green, and blue represent the different subunits of the histone. And this gray part is the DNA wrapped around it, so here's your side view. And then this is, that's funny, I would think of this as straight on, but they're calling it a side view. And I would call that a side view, but they're calling it a top, bottom view. So that's looking at the side of the bagel. And one's looking at the front of the bagel. Who thinks of bagels when they look at histones? Or donuts? Donuts or bagels? Should we take a vote? Donuts? Okay. So that's how it looks. And this is how it assembles, and you do not need to memorize this, but sometimes having a little bit more detail is helpful in understanding it. So you have this H3H4 that assembles into a dimer, and then the dimers assemble into a tetramer. Dimer means 2, tetramer means 4. And then they bind to the DNA. And then H2A and H2B will form a dimer, which will then form a tetramer, and then it will assemble with the H3H4 tetramer bound to the DNA. And that's how it assembles. And so I want you to, so I put this in for you. You asked me the question about the DNA the other day, or you? Oh no, you guys look alike. Who was in recitation on Monday? Of you two. You guys could be brothers. Sorry.
I don't see it.
This is this is the problem that you have as a professor. You've got like a million people in front of you, and they all start to look alike. Thank God for that little diversity. Okay. Question about the DNA you were asking, right? About how is the DNA structure affected by wrapping around? And actually, they had a nice thing in the textbook about this, about how well, mostly just how. Though most of the interactions are with the backbone of the DNA, some of the interactions, there are some bases in the minor groove, and we'll talk more about the minor groove, but that's this part of the helix, this small groove there, that are preferred because it can compact more easily and maintain its structure. So I did get a question in recitation about how the structure of the DNA is affected. If the structure of the helix changes when it wraps around the histone, and I was like, I guess so, but I have no idea. But there was something, there was a nice figure and a little paragraph about it in the book. It was useful. Okay, so I want you guys to notice, sorry if I embarrassed you guys by calling you out on how you look. Just brothers, cousins, perhaps, I don't know, same color hair. Notice how histone and terminal tails protrude from the DNA histone core. Has everyone noticed that? The other sticking out? See these things? All sticking out? Why is this? What is the purpose of these protruding tails? Yes? Okay, so to attach different proteins, and that's one thing, yes, but what else happens on these tails? Yes? Post-translational modifications, right. And some of the proteins that do some of these post-translational modifications are HATs, histone-acetyltransferases, which are going to transfer acetyl groups. Right. And so what's the purpose of these post-translational modifications? Yes?
Well, they regulate what's going on. I mean, they can start transcribing a certain gene or wind everything up so that nothing can be transcribed.
Right, so they're involved in regulation of chromatin structure and function. They might recruit things to help with transcription happening, like they might help to recruit a chromatin remodeling complex, a nucleosome remodeling complex, which will move your histone out of the way and allow access to a promoter. They might recruit other proteins that are going to be involved in tightening up the binding of the chromatin, and so that there will be no gene expression, you'll have repression. So you have to think about, when I talk about how dynamic the chromatin is, I'm talking about how things are moving around and constantly changing and affecting gene expression. A lot of this is regulated by modification of these histone tails to create binding sites to recruit other proteins to do things. Big picture. Binding sites, recruit other proteins, do things. Yep.
Do the tails also help the histones bind closer to one another?
Yes. Do the tails also allow histones to bind closer to each other? Yes, they are also involved in the histones binding to other histones and that tight packing. Yes. Any other questions? Yes.
Quiz I got it wrong because it was like post-translational or post-transcriptional modification. Is post-translational because most proteins when they come in, they come back in?
Yep, so the difference between post-translational and post-transcriptional modification is just what is it happening. If it's post-transcriptional, it means the transcript is somehow being modified, the RNA is somehow being modified. Post-translational modification means that the protein, so the product of translation is a protein, and that's what's being modified. So that was actually, I put those kind of answers in just to make sure people, because people always confuse transcription and translation. And so I want to make sure everyone understands that transcription is getting the RNA and translation is getting the protein. And if you have post-translational modification, that means the protein is being modified post-translationally. You can't modify a protein post-transcriptionally because there's no protein yet. It's RNA. Good question, because I bet you'll half this class had it. Thank you. Okay, any questions? Okay, so this, is this the question you were talking about? I purposely put that in there. Okay, the structure, okay, so you guys, if you have your A, B, C, D, E, F, G's, fine, pull them out. The structure and function of chromatin is regulated by, of course, I kind of just told you the answer, but feel free to put what you like. We can fight about it. Do you want to fight about it? No. I love a good argument. I like to be challenged. Why not? So I'm seeing lots of B's. Great. Post-translational modification of histones. Right. Structure and function of chromatin is regulated by the post-translational modification of histones. And that takes us to this figure, right? So you can see here it's showing each histone subunit here and its N-terminal tail, and it's showing the different amino acids that are modified. A post-translational modification means an amino acid is somehow modified by the addition of either a protein or a molecule. Yes?
Why isn't it regulated by the DNA sequence?
Chromatin structure and function. Why isn't it regulated by the DNA sequence? Well, because I'm trying to think if there's any way that it could be, but because chromatin structure, where are you there? Chromatin structure and function is really about where your histones are and on your DNA and how the histones are interacting with each other. So whether or not you're going to have a more open DNA structure and a more closed DNA structure. You could say the DNA sequence might have some effect because in a roundabout way, if you move, I don't know. Yeah, I'd stick with post-translational. Yeah, DNA sequence is more about regulating which gene is being expressed. It's all together. It's all part of the big picture of gene expression. But when we're talking about the big picture of the chromatin structure, whether things are open or closed, really has to do with where those histones are on your chromatin, and that's very much regulated by post-translational modification of histone tails. Is that satisfying? No. Okay, questions? Yes?
The tails regulate where, you just said something that confused me. The tails regulate how it's going to be wound around?
The tails very much regulate the location of nucleosomes on the DNA in many ways, or how open or how closed the DNA is going to be. Because tails, and what we'll see as we move into this, is the modification of the tails are creating binding sites for proteins. And those proteins are going to recruit other proteins that are going to do things. And some of those things are going to be to move nucleosomes around. Some of those things are going to be loosen up the wrapping of the DNA around. So, yes?
Okay, this might be coming down, but are nucleosomes and histones the same thing?
Yeah, so histones are the proteins that the DNA wraps around. The nucleosome is the DNA wrapped around the histone.
Okay, so the nucleosome is the histone plus the DNA.
Nucleosome is histone plus DNA. I tend to kind of use them interchangeably because nucleosomes are being shifted around. The histones are being shifted around on the DNA, so they're often staying in contact. But sometimes you have more removal, yeah.
So during the nucleosome remodeling, are they just kind of rolling along the DNA, or do they detach and then going back up?
You know, there's multiple models. I'm not quite sure. So the question was, with nucleosome remodeling, are the nucleosomes being rolled around on the DNA, kind of like a yo-yo, you can imagine, or are they being disassembled and reassembled? And I've seen both models, and I'm not sure if it's definitive what's happening, but depending on what textbook you look at, I've seen both models. Okay. Questions? I'm getting great questions. Hopefully that means people are engaged. Yes?
Quick question. I understand that there's like a big picture between chromosome structure and function, but how in depth do you want us to know specifically what affects the structure of this function?
Well, as we go through this, you'll start to see. Because right now this is overview. So I'm being very general. And I think you need to understand the big picture before we can dive into the details. I think you'll start to see as we move on, and when certain things come up that seem very specific to you, raise your hand and say, do you want us to understand this specific detail? And I might be able to say, no, but I want you to understand the function of this and regulation of that. You know, so I can help you to start to gauge the level of detail you need to know.
So that will be more specific as to what the big picture is. Because like, you keep referring to the big picture, but like how broad versus how specific are we supposed to be?
You'll every time you see an instance where you're not sure, just ask me, because you'll feel it out as it goes along. And if you're like, I need to know, does it bind to this DNA sequence? No, too specific. I need to, but if like, so I might talk about HDACs as a whole class, of enzymes, and so I want you to understand that function of HDACs in regulation of chromatin structure and regulation of gene expression. But I might not want you to know the individual names of HDACs and how they bind and how, life is too short. Just don't have time memorizing them. Okay, but please ask every time something comes up. Yes? We're not talking about anything. Yes. We will go into that a little bit. Yeah. Okay. I know sometimes I'm just like feeling like we should do nothing and just sit here and have donuts. Yeah. We're talking about histone. We should have food-themed lectures, right? Okay, let's go on before I waste all of today. Okay, so what is post-translational modification? What does it do? So we talked briefly about it, but I really want people to understand it because I myself am a lover of post-translational modification. I work on ubiquitin. I work on everything ubiquitin related, not everything, but I work on a few ubiquitin related things, and that is a post-translational modification. So you best understand post-translational modification if you're going to be in one of my classes. So I know. Okay, so we spoke about it has to do with modifying proteins, but what is the function? What does it do? What is the point? Like we talked about phosphorylation and signaling. We talked about acetylation with histone. What is the purpose of modifying a protein? Yes?
It alters their shape so they can form a new function.
It alters their shape so they can have a new function. So it's a way of regulating, that's absolutely right. It's a way of regulating the function of a protein. Because don't forget, proteins have jobs. And the way they do their jobs is to usually interact with other things, other proteins, DNA, whatever. And it's all regulated. It's not just random. And it's regulated by the ability of these interactions to happen. So post-translational modification is going to change the shape of a protein and either allow or not allow a protein to interact. So anyone who's had me for 408 has had this drilled into their head. So hopefully they are like, oh, totally, I get this. But it's possible that you don't, and then you'll get it this time around. Okay, so good. So when we talk about post-translational modification of histone tails, we're talking about modifying the histone tail, and that's changing the shape of a histone tail and allow for recruitment of some other protein that only binds the modified form of the histone. Right, because remember how enzymes, remember that lock and key model that you all learned in high school probably? You know, it's like an enzyme active site only fits a certain substrate and it only works that way. So you can think about modification in that way, that proteins and proteins are interacting and they only have, each protein has a binding site for another protein, and if you stick a big phosphate group in the way, it might affect the binding site. It might make it stronger, it might make it weaker. Okay? Okay, so let's keep going. Okay, so specific post-translational modifications create or remove, depending on the modification and the function of a protein, they create or remove protein binding sites. Phosphorylated tyrosine. So if you have a protein with a tyrosine amino acid, you know, tyrosine is an amino acid, it's present in a lot of proteins. Tyrosine can be phosphorylated. Phosphorylated tyrosines tend to bind to SH2 domain containing proteins. These are proteins that have a functional unit on it that likes to bind to phosphotyrosine. When that tyrosine is not phosphorylated, the SH2 domain containing protein won't bind to it. And so this is what I'm showing you here. Here's your protein with your tyrosine sticking out. Your protein would be underneath. Tyrosine, phosphate, and then it's going to allow some other protein to bind here.
So,
Yes?
so if it's missing that phosphate, it won't bind.
If it's missing that phosphate, it won't bind. There's a level of regulation there. Yep.
Now, what if there's two phosphates?
You cannot double phosphorylate a tyrosine. Only one phosphate group can be added. Yeah. But you will have sometimes, you will have multiple phosphorylations on a protein that might specifically create a binding site for specific proteins. Yes?
So is it something specific that we should remember that?
Yes. Because this is key to understanding chromatin structure and function. Now, not specifically phosphotyrosine binds to SH2 domains because that, well, that's really more for 4A when we talk about signaling. But the concept of post-translational modifications, changing the shape of a protein, and that will result in interaction with different domains of different proteins in a regulated way. So the examples that you should understand for the purpose of this class are acetylated lysine and methylated lysine, because histone tails are frequently acetylated or methylated, and that's what regulates the chromatin structure and function. And so when you have an acetylated lysine, so here's your lysine on your protein, which would be your histone tail, right? So just imagine that these words are your histone tail, and you've got your lysine there, and it gets acetylated. It will then tend to bind to a protein that contains a functional region called a bromodomain. Bromodomains bind to acetylated lysines. Don't worry, we'll get further into this. Methylated lysines bind to chromodomain containing proteins. Why are these things called weird things like chromodomains and bromodomains? Well, because someone said that was a good idea. Yes?
So all three of the examples are protein binding sites?
Huh?
All three of the examples.
All three examples are protein binding sites, but they're different based on the modification. Right? So you've got phosphorylation, acetylation, and methylation. Those are three different post-translational modifications. That means that an amino acid on a protein is modified in some way, either by addition of a phosphate group, addition of an acetyl group, an addition of a methyl group, right onto the amino acid. Changes the shape, right? Does that make sense? Would a phosphate change the shape of a protein? Yeah. Would it change the charge? Probably, right? Okay. I guess we're done. It doesn't have to be over. Can we stay here forever? Okay. We'll finish this on Friday. Let me just turn this off.